Greetings, everyone. So in this video, I'm going to show you guys how to handle gene sequence, how to handle sequencing files, and take sequence reads and get a consensus gene sequence from that read that you can use downstream for phylogenetic analysis or other investigations. So a quick background. Gene sequencing analysis or PCR sequencing analysis is basically a post-PCR step or a downstream PCR process. What I mean by that is one would conduct a PCR to amplify a gene of interest or a region of interest, run it on an agarose gel to confirm that the PCR was successful, assess whether the controls substantiate the products, and check if the product quality is good. By that, I mean your positive control has positive amplification, your no template control and negative controls are clean, and the products that you got are stable. So there's no sign of smearing and you have an intense single band for your product. So after those have checked out, samples can be sent for sequencing. And there are many different types of sequencing reactions. In our lab, the most commonly tasked sequencing reaction is Sanger sequencing. And we normally send our samples out to industry to perform the sequencing reaction for us. When we get our sequencing reactions back, it's not in the form of a page with sequences written on it. It's generally in the form of a file that gets emailed in a .ab1 format. And it is these files that contain our gene sequence information. So when you're analyzing gene sequence reads, there's many different types of software available. In this video, I'm going to be using Chromas to process the chromatograms and work with the .ab1 files. I'll be using BioEdit to help with the FASTA files and specifically MegaX to deal with the construction of a reverse complement of our reverse sequence and assist in phylogeny and videos to come. So in this example, we're going to be working with a cytochrome oxidase product. Cytochrome oxidase PCR yielded a 650 odd base pair product, and this product was sent for sequencing, and we have got the sequencing products back, the sequencing reads back. So when we send off products for sequencing, PCR products for sequencing, we are sending a double-stranded DNA molecule for sequencing to basically expose what nucleotides make up that fragment that we have amplified. And depending on the type of sequencing you use, depending on your product size that you want to get sequenced, it may be done slightly different. Different sequencing reactions are more accurate than others. They can handle longer reads than others. And because of that, they get handled a bit differently. So this gene, we did a two-way sequencing. We did a forward sequence and a reverse sequence. Forward sequence and reverse is basically done so that in our double-stranded DNA product, we cover all the nucleotides that would have been incorporated into this product growing from the forward end where the forward primer led amplification, and from the reverse end where the reverse primer led amplification. So the larger the product, sometimes people may choose to include more than one primer to designate where the sequencing reaction would take place. So they would include a forward, reverse, and they may include a primer within the sensor just to get a more reliable coverage, just to ensure that all the nucleotides were incorporated, were sequenced and read properly, and just helps improve the accuracy of the read. because as the longer the product, the longer the sequence read, certain sequencing methods can reach their limitation for reliability thereafter. So our product is 650 odd base pairs, and we got the forward and reverse sequence. So now in this example, we are going to be tasked to take the .ab1 files that have our chromatograms and the nucleotidome sequencing, and actually process that to get a single file that has the nucleotides that the PCR product contained, and it must be in the 5' to 3' direction, which is the standard for many molecular applications. So as I said before, we're going to be using 3 programs for this. All programs are free and readily available. So the programs are ChroMass, BioEdit, and MegaX. So these are .ab1 files, and computers can't handle or can't read these files natively or innately. So that's why we rely on software to help us take these specific formats, analyze them, and get them familiar to the machine, and so we can work with them as well. So here's our sequence files. We got the HCO, we got the LCO read. I'm going to start with the LCO read, and we open these files using a program called ChroMass. So you can download ChroMass and install it. And to open these files, we right-click, open with, choose another application. We should find ChroMass there. However, if it is installed, you can just open ChroMass and go to, these files are stored on my desktop, so I can just navigate to my desktop and open the file of interest. So when we open ChroMass, what we'll see 2 things in particularly. First, which is most obvious, we will see a spectra. And these spectra has many different peaks with different colors, and this is called a chromatogram. So chromatogram has many peaks. The stronger, the higher the peak, the more confident the machine was able to assign a certain nucleotide. They're also color coded. So in this example here, we have a cytosine, which is blue, and other nucleotides assigned in different colors just to help us differentiate which peak corresponds to which. Now, within this, the second thing we will see is in the top, we have a row of letters. These letters correspond to a certain peak. If we put our mouse cursor below a letter and click, we will see a line that is generated, and that line would link a letter to the apex of a certain peak. So each letter is assigned to each peak, or each peak is due to a corresponding letter, and that is how the letters were assigned. That's how our nucleotides were assigned. Now, since this is being used to assign nucleotide positions in a PCR product that was sequenced, there will be regions of noise, which should be expected, and there will also be regions where we get very good, clear, distinct reads. And these are regions that we ideally want because the assignment of nucleotides are the most accurate within these regions. So generally in these AB files, in the ends of the sequence, at the beginning and at the end, we will find a fair degree of noise. And these regions of noise are generally characterized by having messy peaks that do not have a proper shape, and the assignment of letters being unreliable. So instead of us seeing A's, T's, C's, and G's for nucleotides, what we'll actually see is letters like W, R, and Y. So as a good rule of thumb, what we do is at the beginning of our sequence, we would delete nucleotides that we cannot get reliable reads from. So in this AB file, I have deleted the nucleotides that correspond to this region, but I feel that there's a more high degree of certainty that is required. So in this example, I'll just be a bit more strict with that, and I will remove a bit more, bases than what I would need to. So I would try and clean up the regions at the beginning. I'll go to the end. When I'm at the end, I can see the peaks are fairly good. They're not the best, but they're decent enough for me to say that I can trust I can say with a fair degree of confidence that this should be fine to include in downstream work. Once I check both ends that the sequence reads are reliable or good, or at least good enough for taking further processing, I will then go through from the beginning or from which either end you prefer and look base by base for nucleotides or for reads that are not A, T's, C's, or G's. And here's a prime example at 21 base pairs or 21 nucleotides, there is an S. And if I go down, if I click just below the S and I see this dark line over here, I can see that this S corresponds to a small peak, and that peak is supposed to be a guanine. I know this because it has the same color as the rest of the G's. Even though the peak is a bit faint, we can assign a nucleotide to it. Now, what we would then do is continue going through the strand and look for any other deviances that is not an A, T, C, or A, G. Okay, so I have previously done some work on this file, so I'm fairly confident that the sequence reads are good. And what I'm going to do is I'm going to export the sequences from this file onto my desktop, and I'm going to create a new folder where all the files will be stored in. So I'm going to call this folder fly cytochrome oxidase and gene sequence analysis. And in this file, I'm going to rename this as fly LCO, or you can rename it as fly forward or whatever the designation was and the read. And this file is going to be saved as a FASTA file or in FASTA format, and most files will be stored in this format as well. So once that's done, I can close. I generally, you don't need to save this, but if you already have a copy of your chromatogram, you can save this as a reference of where you were, or always back up your work regardless. But if you already have a backup and you already have worked with this, you don't really need to. So I'm going to save it just to be for demonstration purposes. So once we have now done with processing our first sequence, now we're going to work with the second sequence. So this sequence has been processed similarly. I've removed some noise at the beginning, but I am still not happy with the overlap that's taking place over here. So I would delete a few more nucleotides till it enters into a region where I can say with a bit more confidence where the nucleotides were assigned. That was at the beginning. As I go to the end, I can see the end is a bit more stable. Not the best, but decent. And then I would go from the beginning and look for anything that stands out. So right here, we have two Ys. The first Y has the same color as an A, so I'm going to just select it and type A. The second Y, I am going to call it supposed to be a G, so I'm going to assign it as a G. And then I'm going to continue going through the sequence file. And I'm happy with how the sequence looks. I couldn't detect any aberrations when you're doing this. You generally would take a lot of time going through this. This is a sequence that I have worked on before, so most of the aberrations, if not all of them, have been found and altered. But generally, when you're doing this, it does take quite a lot of time because going nucleotide by nucleotide, it can, yeah, it really does require a lot of attention. And how this file gets processed will determine how, will also determine how your corresponding sequences will also be interpreted and processed. So once we have worked with the reverse sequence, we can export this. We can export this similarly. I keep all these files in the same folder on my desktop, and I'm going to rename this as HCO or fly HCO, and I will save it as a FASTA format. I'm going to save this file so I know what has edited. I'm going to close. And now if we go into our fly cytochrome oxidase gene sequence analysis folder, we have the LCO, we have the HCO. Now, both of these are saved in FASTA format. And what we're going to do is we're going to now, these are the sequence reads that are processed. So we're not working with the chromatograms anymore. We just have the nucleotide sequence. So now what we're going to do is we're going to, instead of having two separate files, we're going to merge these files together and try and only work with a single file. So we do that by using a program called BioEdit. So I'm going to open the LCO first. I'm going to open it with BioEdit. So if your icons are highlighted like this, it means BioEdit has recognized that this is a file format that it can work with. If it's not like that, you can just go into your menu and find BioEdit, open it, click open a file. Since everything's stored on my desktop, it is easy to navigate to, and open the LCO. Right? And here we can rename the file, so the sequence. So we rename it by either selecting it once and then selecting it again and editing it, or we can double click it, and under name we can highlight it and change the name to fly LCO read. So you can name all this as forward read or reverse read or whichever primer you use. I find it more comfortable labeling it as the primer set that was used, the LCO and HCO. I'm going to click apply and close. That's the fly LCO. Next, what we're going to do is we are going to open the HCO. And again, I'm going to relabel this as fly HCO, just so it's a bit more pleasing to the eyes and easier to work with. And I'm going to save this file. What I'm going to do now is I'm going to save both files. And what I'm going to do is I'm going to copy the fly HCO to the LCO folders. So we're going to do that by selecting the HCO just with a single click. Go into edit, select copy sequences to clipboard. The emphasis is on copy to clipboard and in FASTA format. Then select the window that has the LCO read. go to File, Import from Clipboard, and you will find that now you have one file that has both your LCO and HCO read. If you are working this with another gene, then it will have your forward and reverse. So now the next thing that we need to do is save this file as a single file in FASTA format that has both of them labels. So this will be our fly LCO and HCO. So we label this with fly LCO and HCO, so we know straight off the bat that this folder has both sequences in it. So by labeling it more efficiently, it just helps with our workflow much better, just in case we need to double check or in case we are looking for something. So once that is saved, we can close BioEdit. And in the same folder on our desktop, we can work with the fly LCO and HCO separately. So this one folder has the culmination of our two AB files of the individual process FASTA files all in one. So now our HCO is the reverse, basically the reverse sequence of this gene. And what we need to do is, since it is the reverse sequence, we need to reorient it so it's in the same direction as our forward sequence, as our forward strand. And since it is the reverse sequence, it is the bottom, is on the bottom strand of our PCR product, which means it is the complement of the forward sequence. So we need to get the complement of the complement so they are both in the correct orientation and direction. We do this by using a software known as Mega. So we just go to Mega, click the file to open it, to open the application, then go to File, open a session, and navigate to our folder on the desktop where everything was stored. Click Open. Mega will ask you whether you want to analyze or align. Click Align. And then what you will do is you will see that both sequences are present, and where you have made any alterations will be present as well. Now, this is our reverse sequence. What we want to do is reverse the sequence and complement it. We do that by right-clicking our reverse sequence, our HCO, and clicking reverse complement. And when we do that, the orientation of this sequence should be the same orientation and direction as our forward sequence. Once that is done, we can select both and align them to see where they overlap, just to help us process them a bit better. So we'll select both, we'll go to alignment, we'll click align by muscle and click OK. And what we will find is there will be a long stretch where both sequences, both the forward and reverse, match each other completely. And there will also be regions where the reverse sequence or the forward sequence are out of this symmetrical image. We call these the overhangs. So these regions where they overhang, many people would argue that because they are overhanging, they are not part of this region where both sequences complement each other, so we cannot rely on them. One could argue in response to that since the chromatograms had distinct peaks, we can use these regions with a fair degree of confidence that they should be fine. If the chromatogram peaks for these nucleotides were questionable, then yes, you should not have included them in the first place. But if you process them properly, you can work with this with a fair degree of confidence. So once these files are aligned, remember, both of these are now in the same direction and orientation. And since one read was from the forward and one was from the reverse, they will naturally have these gaps present. So what we are going to do from here is we're going to export this alignment as a FASTA format into the same folder on our desktop, and we're going to label this as aligned fly LCO and HCO. And it is in FASTA format, which is what we want, and we can click save. Once we do that, we can close Mega, and we can look for our file that we have just saved. In our instance, the folder automatically updated and the aligned file is right on top. Now that we have both sequences, the forward and reverse, aligned to each other, and we can see where they overlap, we can see that they match well, we now need to generate a consensus sequence. A consensus sequence is basically a sequence generated by analyzing the complementarity or the similarity between the forward and reverse sequence. Basically, if they are aligned and they are identical, then the nucleotides from both of them would be reflective of what the nucleotide sequence of your product was. And we do that but in bioedit. So we can right click this file and we can either open it with bioedit like this, or we can just go to bioedit, open folder, and open our aligned sequences that way. Now, the orientation of how the LCO and HCO would be presented will be in the same orientation as we saw it in Mega. And if we see something different, then it's most likely because the file wasn't saved or the file got corrupted. So now that we can see that the forward and reverse sequences are still aligned, we need to generate this consensus sequence. We do that by selecting both reads or both sequences, and we do that by holding Shift and clicking the reads that we are interested in. So in this case, it's just the LCO and HCO. We then go to alignment. We click create consensus, and bioedit automatically looks at each sequence and sees what nucleotides are present, and then just drops them down into the consensus sequence. And we can see that the consensus sequence is in line with the maximum length of our alignments. So we know that no strange anomalies has occurred due to software related errors or anything like that. So now this consensus sequence is very important. Any further processing we do with this gene sequence or with this nucleotide sequence will be based on this consensus. It is the culmination of both the forward and reverse. That is the short and simple definition of this consensus. So what we're going to do is we are going to rename this as fly cytochrome oxidase cytochrome oxidase PCR, or we could just name this in a bit more descriptive, presumptive green Boswell fly. And this consensus sequence, we are going to save separately from these two files. So we have a single file with our consensus sequence. We do that by copying to clipboard our consensus file and saving it in a new bioedit window. Now this window we're going to save. We're going to save it as a FASTA format, and we are just going to save it as a green fly cytochrome oxidase. Partial gene sequence. Once that is done, we can close BioEdit. If you want, we can save this. There's really up to you. And now we have a single file that has our cytochrome oxidase of our fly species. It's presumptively a green bottle fly, and this gene sequence would basically be reflective of its partial cytochrome oxidase subunit 1 gene sequence. So that is how we go from our AB files to a consensus sequence, which has the resultant of both sequencing reactions stored in one file. If you have any questions, do let me know. Keep well, take care, bye.